Hello everyone. In this session, I am going to discuss the toxic impacts of dioxin on liver of albino male rats. This is a study conducted in the Department of Pathology and Poultry Diseases, College of Veterinary Medicine, University of Diyala, Iraq. The aim of the study was to determine the effectiveness of 2378 tetrachlorodibenzodioxin, or commonly called as TCDD, in imparting toxigenicity in the liver cells of albino male rats. So this dioxin compound, this TCDD, it is a very toxic chemical compound, and it is commonly found in the manufacturing of technical chlorophenols and other derivatives such as herbicides, 2,4,5-D, 2,4,5-D, etc. It is also released to the environment by the burning of food crops, trees, and other things, and it is also released by incomplete burning of organic compounds. So this is very toxic and hazardous because they have an ability to persist in the environment or the atmosphere for long periods. So how this compound is toxic to the mammals? So in the case of mammals, there is a aryl hydrocarbon receptor of the dioxin receptor, and it is a ligand activated transcription factor that is leading to TCDD toxigenicity, and it is also involved in the pathogenesis. So when this dioxin they enter via the food or inhalation into the body, they will reach to the circulatory system, and then from the circulatory system they will enter into fat and liver cells. So inside these cells, as we said before, there is a aryl hydrocarbon or the AH receptor. Okay, this will tie them to a secondary protein receptor called nuclear translocator, and this will tie them to a DNA strand, and this will diminish the immune system function, and it will cause into the tissue disruption. So due to this reason, it is considered as a group 1 carcinogenic compound by the International Agency of Research on Cancer. So that is about the toxigenicity and the introduction about dioxin. Now we will discuss the materials and methods required for this experiment. So 30 albino male rats with ages 4 months and body weight 210 to 260 gram were taken for this experiment. They were given 12 hours of light-dark cycle at 21 degrees Celsius ambient temperature. Diet and drinking water were also given. So the TCD stock solution for administrating into this rats were made, and it was dissolved in acetone freshly dissolved in corn oil, and then this acetone was evaporated under nitrogen. So this is how the TCDD was made for administration into this rats. Now the 30 groups rats, they were divided into 3 groups, each containing 10. Okay, and the first group, that is called the control group, these rats received normal rat pellets. The second group, that is acute stage group, they received 125 gram of TCDD for 30 days. Now the third group, that is the chronic stage group, they received 125 gram of TCDD for 90 days. All these animals, they were sacrificed for the biochemical and pathological analysis at 30 days and 90 days of the experiment. For the biochemical analysis, fasting blood heart was used to determine, and it was collected by cardiac puncture technique, and it was collected into the test tube, and it was left for 50 minutes to stand, and then it was coagulated in the refrigerator at 500 rpm for 50 minutes in the centrifuge, and the serum was separated, and it was kept at minus 20 degrees Celsius for the analysis. 3 biochemical tests were done. The first one was to determine the antioxidant enzyme serum, that is peroxynitrite radical concentration. The second was to determine the triglycerides or the high density lipoprotein HDL. It was done by commercial kits. And the third one is to determine LDL and VLDL cholesterol concentration, and the fourth one is to determine the serum catalase activity. For the pathological analysis at the 30 days and 90 days of experiment, all animals in the 3 groups, they were sacrificed by longitudinal abdominal opening. Then the liver was carefully dissected, and it was seen to record any abnormal in size or color or consistency or adhesion or so they were immediately fixed in 10% neutral buffer formalin and graded with ethanol, that is 70, 80 or 90 and 100% ethanol, and they were embedded in the paraffin and stained by routine stain. Now we will discuss the results of the experiment. The antioxidant enzyme serum peroxynitrite radical concentration, they decreased significantly at the group 2 and group 3 when compared with the control group, and they showed highly significant decrease at the 90 days. In the lipid profile, HDL showed significant decrease in the first and second group, mostly at the second group in the 90th day, while they showed significant increase in LDL and VLDL, mostly at the 90 days of the experiment. The group 2 and 3, they observed significant decrease in the level of catalase activity also at the 90 days of experiment. Now the results of the pathological analysis. In the pathological analysis, the first group, they didn't show any change. The second group on the 30th day, they showed zonal or lobular pattern pale in the serosal surface with massive areas of necrosis on the surface. In the color, and it may be hepatitis. In third group at the 90th day, they showed liver grossly showed the multifocal areas randomly distributed of necrosis and abscess with jaundice. Hepatitis abscess with nodular hyperplasia of liver parenchyma and fatty change with hard spot of calcification was also seen. All the liver parenchyma seen, they consist of multiple granulomatous lesion causing the hepatocytes atrophy and liver damage surrounded by variable size of numerous giant cells with center of dystrophic calcification. The analysis of the results. The liver is the major organ for this toxicity of TCDD, and it is considered to be the principal target of toxicity of dioxin. So the dioxin is affecting the liver cells mainly. Then the decreased level of the catalase, which we see in the second and the third group, specifically at the 90th day, due to the reactive oxygen species, is done through bioactivation of benzene group, and they also play a crucial role in the toxicity of TCDD. The liver damage that occurred due to this TCDD effect, and it was indicated by the granulomatous change at the 90th day, and it was also determined by the inability of the liver to metabolize and detoxification of lipid. The pathological changes indicated in the current study that TCDD is always dose-related response. So the second and third group, they revealed pathological changes, and mostly the third group that was mostly damaged, because then they were characterized by hepatic damage, enlarged, and jaundice, centrilobular vacuolated, and other kinds of damages. So from this experiment, we found that how the TCDD, they are being very hazardous to the mammals, including humans, and definitely they have to be considered as a carcinogenic agent.